although this one follows up more closely on what David was talking about last week with the proteomics. So this week we're going to talk about gene expression. And we're going to go over microarrays primarily today, although a lot of these principles apply in general to gene expression and to RNA-seq and also to proteomics and a lot of other situations where you're mining data from different samples that is high dimensional and highly variable. We talk about data formats, QC, normalization, differential expression, which is usually the focus of interest for most biology labs, functional enrichment, databases, and I'm also going to offer a small intro to R in the sort of after the lecture segment, because my assignments today are in R rather than Python, because it's traditional to do microarray work in R. Yes? Yes? Unfortunately, David controls the IBB website, and he is incommunicado today, probably on a plane somewhere. So they'll have to wait till tomorrow. I mean, I could give them to you after class if you want. I have them on my little flash right here. But we will upload them tomorrow. All right, so this is my non-molecular biologist slide, but it's relevant because we were talking about proteomics last time. And today we're going to be looking at microarrays, which measure RNA. And we have this nice little graphic of the central dogma of molecular biology, just to keep in mind that when you measure RNA, we call that gene expression, although in fact it's in the middle of a pipeline where there's all kinds of control steps upstream of the RNA that are involved with translation and epigenetics, chromatin, et cetera. Then there's the level of transcription. And then there's another RNA modulating step, which is the stability of the RNA molecules. So when you're doing a microarray or an RNA-seq, you're measuring a snapshot of the steady state RNA levels, more or less. And you don't get a complete dynamic picture of how the RNA is actually being transcribed, nor how it's actually being translated. Similarly, when you measure proteomics, you're getting a steady state snapshot of protein contents in the cell, and you're not actually measuring the number of proteins that are currently being translated, because again, there's translational modifications, post-translational modifications. Different proteins have different half-lives in the cell. So each of these technologies provides a view of gene expression, but the whole thing is actually a bit more complicated, and you have to keep in mind these biases, that your technology has these limitations, that you're getting a steady state snapshot. Okay, so we're going to start with microarrays because this was an important technology. We've started to move beyond it, but I want you to understand how it works, and we're going to look at data today that come from microarrays. There's actually quite a few large-scale microarray projects still underway, and there's even new grants being funded to continue projects using this sort of traditional technology. So it's peaked. It may be slightly on the wane, but we're still producing a lot of data, and probably the bolus of publications related to microarrays are still moving through the pipeline. So you're going to continue to see hundreds and thousands of important discoveries made using microarray technology. So what's a microarray? It's actually a simple concept that goes back even further in molecular biology to the dot blot and the northern. You guys must know what those things are, but a northern is when you put RNA into a gel and then probe with a specific sequence. So you're lighting up an RNA molecule, usually size fractionated by electrophoresis. A dot blot tends to be the other way around. You put a single sequence or a series of them on a substrate, usually a filter, and then you probe with a mixture of something else. When we're talking about RNA, so you could probe with labeled RNA that came from some cell, and the different dots light up, measuring amounts of the corresponding sequence in your mixture. So that's what it's about. So in a microarray, the probes are fixed, and the RNA from the cell, the mix of messengers, is the probe. So is labeled material. So when we talk about probe, we're actually talking about something that's fixed on a filter, rather than the more traditional way of thinking of a probe as something you mix up in a tube and hybridize to a filter. So you put the probes on the filter and you label bulk RNA. You make probes for a lot of genes. So your filter contains many, many spots so you can interrogate many different genes at the same time. And so in effect, it's a massively parallel experiment. You make the spots very tiny so that you can fit lots of them onto your array, and so that you don't need so much RNA to hybridize to the array. And the technologies for making the spots smaller and smaller was the main thing driving the development of microarrays over about a decade worth of time. It's sort of currently plateaued because making them smaller doesn't gain anything anymore. They're small enough. We'll see how small in a sec. And then you want to make quantitative measurements, right? You're hybridizing a labeled mixture of RNA to some dots, and you want to know how bright each spot is. In comparison to other spots, but more importantly, in comparison between two different RNA samples that may come from two different kinds of cells. And those types of comparative quantitative analysis is what this lecture is all about. So just the obligatory PubMed slide showing the growth of publications that mention RNA technology from 2000 through 2005, which was kind of the golden period for microarrays. I have some other slides somewhere that shows that after about 2005-06, the number of new publications about RNA, microarray, sort of plateaued. So we're still seeing lots, but we're not seeing this dramatic growth. Now the dramatic growth is in publications that make use of sequencing technologies, which we'll get to next week. So this is the non-micro array. This is an array that you might make by hand in the lab with a few hundred probes, and then hybridized with RNA from some cell sample. It's very typically that the probes were in duplicate. In other words, you're trying to reduce a little bit the variability, but as you could sort of see, there's a lot of physical events going on this piece of filter paper or cellulose or whatever it is that may interfere a little bit with getting a perfectly clean signal. Smudginess, spread of signal from one spot to a nearby spot. Also, that you can also see just by looking at this that you're going to need to do some sort of image analysis. The more automated, the better, to turn these circular blotty things into numbers so that you can start making comparisons and making conclusions about changes in gene expression. So these are challenges that were being faced by the microarray process. So things really took off when people decided to move away from cellulose filters and start spotting DNA directly onto glass, or some variant of glass, which they started to call a DNA chip. The spots could be much smaller. They could treat the glass with some substance that would cause nucleic acids to bind very tightly. And then you could start building automated spotting robots. Okay, so cDNA microarrays came first. You want to interrogate a lot of genes. It actually is quite a bit of work to make probes, but anyway, people said, well, here we know a collection of transcripts or cDNAs from this organism. We go to GenBank, we type in a few keyword searches for our organism and for, say, RefSeq transcripts, and we get a collection of sequences. We either request from scientists that they send us clones for all of these sequences, or we start synthesizing them using our DNA synthesis machines. And in any case, we end up with a bunch of tubes that have cDNA sequences. In order not to use them up so quickly, develop a protocol for taking a tiny aliquot from each of those tubes and then putting it through PCR, maybe cloning it into a vector so that you can use common primers to PCR all of them under a common set of conditions, quantitate, dilute, requantitate until you get tubes that have molar equal amounts of the various genes. Spot them onto the glass slide. Now you want to prepare your sample. RNA from 2 different samples. We'll call them cancer normal, just as an example. In the cDNA example, you tend to label those 2 RNAs in different fluorescent labels, different colored fluorescent dyes, mix them together, hybridize onto your spotted array, and then expose and collect fluorescent image data in those 2 different wavelengths. So you excite and collect in a fluorimeter. Make 2 scans, essentially 1 for each color. Combine the scans and calculate the ratios for each spot. So here's a spotting robot, and here's an example of a spotted glass slide. Ordinary microscope slide may be coated with something to make DNA stick better. And you've got tens of thousands of tiny spots there, roughly the size of a dime. All right, so you label and mix your RNA, hybridize it onto the slide, scan in 2 colors, get 2 images. These are false colors, obviously. Fluorescence is just fluorescence, but you're collecting it at a specific wavelength. Then either, image-wise, just for fun, you can combine the 2 colors, and then mathematically, you're combining the scanned values at the 2 wavelengths for each spot and calculating ratios. Obviously, there's a bunch of issues, computational, mathematical, image analysis issues that are going on here. Because as you can see from real data, the spots are not perfect. They have holes in them, they have edges, they have streaks. Really bright spots send fluorescence to overlap the adjacent really dark spots. So there's a ton of software image analysis issues that go on here. For a while, bioinformaticians were trying to tackle this, but then they realized that image analysis is something that goes beyond just biology. There are entire scientific fields of advanced image analysis and sophisticated software. And more or less, this whole problem of turning spots into numbers was outsourced to the vendors who brought in engineering experts in image analysis. So we don't tend to spend a lot of time in the biology lab tweaking the image analysis of these spotted filters. And in fact, when people were doing this, the more they tweaked the images, the worse their data became. Because you can't consistently tweak the images on every sample the exact same way. So you end up introducing biases by intervening in the image analysis. The more automated the process, the more reproducible it becomes, and the more computationally valid your comparisons are between your treated and your control. Okay, so data acquisition involves scanning the arrays, quantitating the spots, background subtraction, some sort of normalization, because the fluorescent units that's being measured by your fluorimeter may be different for the two different dyes. So you're not getting molar equivalent brightness for the same amount of hybridization. So there needs to be some sort of standard and some sort of normalization. And then you need to normalize across all your samples, because your dyes may not perform the exact same way when you do the experiment on different days. There's a million variables that go on: the age of the buffers, the temperature in the room, the voltage that the laser that's exciting the fluorescent image, the fluorescent molecules, the exact calibration and focus of the fluorescent detector. There's a lot of variables that go on. Normalization accounts for most of those, because you can compare the brightness of one dot with the average brightness of all the dots that were collected on that filter in that pass of the scanner. Then there's other normalization methods that come into play when you're trying to work with a batch of similar data. I'm going to cover that in a couple minutes. But in any case, you're exporting a table of fluorescent intensities for each gene in the array. A company called Affymetrix got into this business around year 2000. And they had a number of important innovations, but probably the biggest innovation was just black boxing all of the data collection method, turning it into proprietary software that the investigator could not tweak. And this allowed people to take sample, put it in one end of the system, and get a table of numbers out the other and move on to publication, rather than spending months or years trying to optimize every one of these bullet points for themselves. And the nice thing about the Affymetrix system was that it was highly reproducible. If you put the same sample on two different chips, even in two different labs, you would get 97, 98% correlated intensity results for each of the genes on your chip. So if the same sample measured twice give you pretty much the same data, which was not the case for the hand-built two-color arrays. So how does the Affyarray work? Instead of whole cDNAs, cloned and stored in the freezer, they used synthesized 25 base oligonucleotide probes to represent each gene. Not just one, but a number of oligos. They started out with about 20, then over generations of their chip, fewer and fewer. Current Affy chips usually have 11 probes for each gene, and they're highly 3' biased, which is sort of interesting. But because the protocol they promote involves a poly-A collection cDNA synthesis process, by having 3' bias probes, they get a stronger signal and a little bit more reproducible, because the 5' end of the cDNA synthesis process is a little more inconsistent from one sample to the next. They also had this notion of a perfect match and a mismatch probe, which I'll get to in a second. You can have any number of genes on a chip. I don't know why I say 20,000. They're up to 1 million now. But there aren't 1 million genes for, say, the human genome. And so that capacity isn't really that valuable for a lot of purposes. There are SNP detection chips where 1 million is a good number or 10 million is a good number. But for the gene expression chips, they haven't continued to make them smaller and pack more features on, because they hit a plateau in terms of cost benefit and just return of useful data. The chips are moderately expensive, like in the sort of $300 to $500 per chip. And they're not reusable, and you use a single chip for each sample. You don't combine the treatment and control in a single hybridization. You do a treatment scan and a control scan, and then you combine them at the end in your informatics analysis. Advantage of this is in terms of experimental design, you can have multiple treatments and different controls and sort of ANOVA type combinatorial analyses. And you can decide later which groups of samples you want to compare in your differential expression analysis. You can take your samples and pull some archival data out of someone else's experiment and make a comparison if they have a control that might work for you. The gene chips sort of look like this. They actually end up being about the same size as the microscope slide I showed you, and the active hybridization area stays about the size of a dime. That just seems to be consistent with the way fluids travel in the laboratory, that if you make it much, much smaller, it's hard to get consistent results. Not 11 base pairs, 11 probes of 25 bases each. I'm about to show that now. OK, this is your typical probe. They are 3' biased on the mRNA. They're not necessarily equidistantly spaced. They have some software that tries to choose probes that have a good GC versus AT distribution and that are unique to the RNA in question. You don't want to put a probe in a region that's shared by 20 different genes in a same family, because then you're getting, you're not measuring the expression level of the gene you think you're measuring. So they have some software that tries to optimize the GC content and the uniqueness of each probe, given the constraints of the length and composition of the genes. They also have this thing called a mismatch probe, which is in the center of a 25 base probe, they change one base, and they make that a separate probe on the array. So for every one of these 11 spots, there's a second probe that has one base different in the center. Thermodynamically, 25 bases disrupted in the center should have a much worse binding constraint than a perfectly matched pair of sequences. Empirically, when we observe the behavior of these mismatch probes, they are wildly inconsistent, and they seem to add noise and no value to the data, which is fascinating that they've continued to double the cost of producing the chips for 15 years after it was clearly proven that the mismatch probes subtract value from the data. Yeah, there is. Yeah, most of the analytical methods that I'm going to talk about ignore the mismatch probes. But they're still there in the data file if you want them. And Affy is stubborn about that. At the bottom is a false color image of a hybridization using a bunch of different probes. Initially, they actually did this. They tiled the probes for the same gene next to each other. And so you could just take a little section out of an array and see how well each of the probes hybridized for your gene. And then they had an algorithm that subtracted the perfect match from the mismatch, averaged them together, and gave you a numerical score for the hybridization intensity of that gene across all these probes. I have a better picture here, just because it's messier, which more accurately represents the real data. So this is still a false color synthetic image. It's not an actual picture of the array. It's showing intensity in some kind of a color-coded scale, and the pixels are just a lot smaller than the cells of the array. So this guy down here and this guy here are equivalent measures of the same kind of data. I won't swear that it's the same exact probe set. But darker is less hybridization, so it goes from red to blue to dark blue. Actually, and then really bright is over here. What you can see is a bunch of things. First of all, we have a bunch of probes that are all supposed to be measuring the expression of the same gene. And if you just look across the different probes, they give hugely different performance, from essentially no hybridization to burnout, right? They've exceeded the level of measurement that the system can measure. Different probes for the same gene. So that's not great. And then the comparison between the perfect match and the mismatch is also somewhat inconsistent, very inconsistent. Sometimes the perfect match is better, sometimes the two are similar, occasionally the mismatch is better. Yeah?
But in some matches you can see that perfect match and mismatch quite similar.
Yeah.
So isn't that a good indication that we shouldn't use that?
You could do whatever you want with that data. People have tried a lot of approaches to this. I'm going to show you the approaches that have won out in the evolutionary process of acceptance among biologists and reviewers. So yeah, you could probably come up with some method of gaining information from the mismatch primers, but or excluding information from the mismatch primers. But overall, it turns out to be too inconsistent across all the different genes. And the method that works best, in my opinion, is to ignore them and concentrate on the perfect match probes. I'm going to skip the movie. So this is like a false color image that's collecting data from maybe 100,000 probe sets on an Affy chip. And the point of looking at an image like this is to see if there's any obvious artifacts. Because an Affy chip is a physical device, right? It's a slide that you could hold in your hand, and solutions wash across it. And it could have dust or hair or a clogged well, and or it could be the whole thing is on a slant because you didn't load it into the clip holder properly. And so sometimes you can see obvious left to right, top to bottom scratch artifacts on your Affy chip. A bubble, a thumbprint, a piece of hair. Didn't load hybridization buffer on the right-hand side of the cell, or some reagent spilled. So looking at the false color image can tell you some of that. The real, actual image can tell you more, but we don't store those. We only store the intensity values, and then we recreate the image from the intensity values. But something like this guy over here, where all the right-hand intensity values are lower, that would show up on the recreated false color image. So that's worth doing. I mean, it's a pain if you have 100 chips in your experiment. But given the fact that you're going to try and publish this data, you probably want to validate that all 100 chips don't have obvious artifacts on them. The impact of the artifacts has been reduced quite a bit by randomizing the position of all the probes. There's no inherent reason why the probes need to be next to each other for the same gene, right? You've got 22 probes for each gene. Put them in random locations all over the chip. So if the upper right quadrant of your chip has, lower signal intensities, it won't bias very much the overall gene expression measurements that you make. And if you normalize that chip in a reasonable normalization process, then when you compare it with another normalized chip, you may still get valid data. So the Affy system is automated. It computes a single value for each gene from all the probes, match and mismatch, using kind of math that's not great, but it's highly reproducible. That's the nice thing. Same sample loaded on the same model chip, scanned a different day or in a different lab, is going to give you highly correlated results. Right, talked about that. So your final output, after you get that intensity image, after you combine all the probes, is some kind of a pivot table or Excel table like this, where the rows represent genes and the columns represent different chips or different samples. And this is what you use typically for your input to a wide variety of different gene expression analysis, differential expression software. There's only some of the software that can go back a step, read the image data, and create its own normalized values combining the individual probes. We are going to talk about that as well. One other thing that Affy does that some software use and others don't is they have a pass-fail kind of system for each gene, which they say is P for past, or P for present, A for absent, and M for kind of don't know. It's sort of in the middle. Marginal, that's what they call it, marginal. So you could, in fact, throw away all the ones that Affy calls absent, and there's dozens of papers saying whether you can or cannot mine useful data out of those. No, that it doesn't meet the QC, the internal QC. And the internal QC has to do with a combination of whether the perfect matches are stronger than the mismatches, and whether the overall sort of standard deviation across all the different probes exceeds a certain value, and whether the overall signal across all the probes exceeds a certain value. So they have their own algorithm which changes from version to version of their software, whether a given gene, after combining data from all the probes, meets their QC requirements. Clearly, their absent ones are worse than their present ones. And whether or not you want to use that data or want to throw them away is part of many different analysis pipelines. I've done both, throw them away, not throw them away. Throw them away seems to be smarter. OK, so I threw this in because David showed so many nice plots yesterday. So one thing you can do is just plot two chips against each other, one normal and one hypoxia today, because these are actually part of the data set that I'm going to give you for the homework. Now, when you just plot the intensity scores, you'll notice that the range here goes fairly high up. It goes from 0 to 40,000 in sort of arbitrary fluorescent intensity units measured by the Affy machine. But down here in the low end of the spectrum is where almost all your data is located. And that's for a few reasons. But just in general, gene expression is kind of exponentially distributed. There's a lot of genes that express at low or no levels in each cell, and relatively few genes that express at very high levels. In this view, the distribution of genes that are higher in one sample or lower in the other doesn't seem to be particularly strongly biased. But that's kind of an artifact of the view. And it's not really a very good way of viewing this data. A better way is to log transform the data. This is log base 2 of the same data set. And it spreads it out quite a bit so that you can see the data more. And at the log 2 level, there's a bit more variation, more differentially expressed genes here in sort of the lower half of the data set. So we're going to be talking about log transforms a lot when we talk about array data. But just in general, when the data is exponentially distributed, you want to log transform it to make it more evenly distributed. And clearly, this data set is spread out on the x and y axes better than this one. So you're getting more bang for your buck. You got very little information about all these guys that are packed in down here at the low values. And here we've got more information about these genes. OK, I'm also going to be talking about something they call an MA plot. And there's a few different ways to do this, but very frequently what we want to plot, or what we're interested in, is fold change, right? How much does this gene change from treatment to control? And again, I'm using hypoxia and normal because I got a nice sample data set for you. But it could be cancer and normal, or it could be drug treated and untreated, or cell line and knockout cell line, whatever you want to study. So the m-axis is the fold change axis. So it's just the log of A over B, the change from sample A to sample B for each gene. All of this is calculated for each gene, and you have one spot for each gene on the array. So this is already after we've summarized the probes into one number to represent expression of that gene on that array. The a-axis is intensity, how bright it is. And again, you're interested to know, is the fold change a function of brightness? Do we see more variability, more change in gene expression at genes expressed at high levels, low levels, or somewhere in the middle? Also, in this case, when I do this MVA plot, I'm comparing one hypoxia replicate with another, and then one hypoxia replicate with 3 replicates of the normal samples. Ideally, what you would expect is more fold change between hypoxia and normal, and less fold change between hypoxia and hypoxia. And I sort of kind of see that for some of the normal replicates, but it's not as strong as I might like. So surprisingly, hypoxia doesn't have a huge global effect on gene expression that I can visualize at this really gross level. It's obviously going to have an effect on some genes that are up and some genes that are down, but there's also background variability from one sample to the next. There's a bunch of genes that seem to just sort of randomly, or for reasons that we are not able to experimentally control, change expression from one sample to the next. So these are true biological replicates, and growing cells in cell culture and treating them with hypoxia is not a perfectly consistent art form. And gene expression in general is a highly variable entity to try and capture numerically. So whatever gene that you're trying to study, its change in expression, you're going to have to deal with the fact that there's a lot of noise. There's a lot of other genes that are changing in expression randomly and not in response to the treatment that you're applying.
So A is on the y-axis and M is on the x-axis?
M is y and A is x. Yeah, so intensity goes to the right. As genes express more, it's off to the right.
So what does this actually tell you then? So if you have a low overall intensity.
Well, these are actually post-normalization. What you don't want to see is some curved distribution or a diagonal distribution showing that all of the high intensity genes, at higher intensities, the fold changes go up. That would be bad. So at least in this case, the median runs through what looks like the center of the distribution, and I don't see a huge curving. This is post-normalization, and we're going to talk about normalization methods in a second. So this is more of a QC metric. It's not an exploratory or data analytic method. It's just seeing whether your data distribution looks decent, whether it's symmetrical around the median rather than skewed, and if you could pick up differences between replicates versus differences between treatments. But this is not the best way to visualize that because there's a lot of noise, and there could be really key genes that are showing up in treatment and not changing in control. So you're going to need to look at the per gene level, obviously. OK, so just in general, experimental or hypothesis-driven goals for a microarray experiment typically fall into 3 categories. You want to find the genes that change their expression levels between experiment and control. We're looking for differentially expressed genes. That's the most common experimental design and goal for these kinds of experiments. And it could be qPCR, microarray, RNA-seq, other technologies. You want to do the same thing with proteomics as well. What proteins change levels between experiment and control? Another thing you might want to do is to classify your samples based on gene expression profile. There's a set of genes that consistently go up in cancer versus normal. And we can take an unknown sample and do a gene expression profile, test those gene levels, and put it into bin 1 or bin 2 based on that classifier. That's another approach that's very interesting. Third approach is to find overall patterns in gene expression among, say, groups of biologically related genes. So a particular gene category or pathway is induced by this treatment. Or if you have some sort of interesting experimental design, say a time course over development, you could see a group of genes go up and down together, or at least synchronously, if not at the exact same time. You know, they follow each other across a time course. So that's another form of pattern identification that we might be interested in. So I'm going to address these things sequentially, but I'm going to spend most of my time on differential expression. So our basic data analysis involves looking for fold change, relative increases, setting a cutoff filter for noise, clustering the genes by similar changes, or clustering the samples by similar gene expression profiles. And in fact, all these things can be done simultaneously. And just a general map of experimental approach to gene expression experiment, where you have data, you normalize, and by normalize, I mean essentially get data sets that allow you to measure expression per gene, per sample, in a way that you can compare across samples. Filter, you might want to remove some subset of the data that doesn't meet some level of criteria. We'll revisit that again in RNA-seq. There's measurements that are too low to be reliable. There may be measurements that are too high to be reliable. There may be outliers of one sort or another that for statistical reasons you are justified in removing from your data set in order to make a more accurate comparison between treated and control samples. All right, then we have 3 different, somewhat independent analysis pathways where by looking for significance, we're simply looking for differential expression. By classifying, we're looking for those genes which consistently are different across treatment versus control, which is not exactly the same thing as most differentially expressed. And then clustering is something that you could apply to genes or to samples or both at once, to say these genes change consistently in their expression across this experimental design. And then all 3 of these experimental workflows end up generating gene lists. Lists of differentially expressed genes, lists of genes that make good classifiers, list of genes that fall into clusters. And for all of those cases, you want to go through a step of functional annotation. What do we know about these genes? What pathways, biological systems do they represent? Particularly in the case of differential expression, what biological functions are enriched in this list of genes? Are we seeing, response to stress genes? Are we seeing genes that have previously been identified as associated with my disease of interest? Are we seeing genes from a pathway that we didn't expect? Are we seeing, say, chromatin remodeling genes when we were studying cancer? That would be an interesting and previously unusual and noteworthy observation. So this is our overall framework for doing our data analysis. Just in general, there's a background level of variance in genes. So when you look at one replicate and another replicate for the same treatment, low glucose 1 versus low glucose 2, and you just plot them, this is a log-log plot like I showed you before, there's some variance, but more of the large variance outside of the threefold are down here at the very low levels. What that represents are measurements that are really below the accurate detection level of the system. So, you measure 4 fluorescence units one time, and you measure 2 fluorescence units another time, and that's a twofold change. But it's not the same as a measurement that goes from 2,000 to 4,000, because there's many more data points of fluorescence being observed, and just in general, a measurement that's in the center of the detection bandwidth is going to be much more reproducible. So we discount these very low level fold changes, and we see that it's pretty good. And then when we go low glucose versus high glucose, up here at slightly higher fluorescent levels, we see changes that are bigger. There's more genes that are threefold up and threefold down in response to glucose treatment than they were 2 replicates of the low glucose treatment. So, in a very gross sense, this experiment seems to have worked. We seem to see genes that are responding to our treatment with fold change. Also, given the fact that we looked at this here, the same treatment has a lot of variability inside of this twofold bandwidth. So we probably want to ignore all those genes that are differential inside that twofold bandwidth, because they'd be different anyway, even if we hadn't put the treatment on. So it's a little bit of data exploration. There's also this idea of, well, these are cell culture yeast, I think. These are mice. And the variability that you observe in a mouse is a lot more. So these are technical variants. So it's the same liver sample put on the array twice. And now we see what's more of a classic volcano plot, meaning more of the variability is at the very low levels. And these are the twofold window, twofold up and twofold down. So we would want to ignore anything inside of these red lines. This guy here, these are samples. These are 2 different mice, still with the same treatment, but now we've got additional biological variability thrown in. And you can see that there's actually quite a few genes outside of those twofold bars. So that tells you that if you want to pick up treatments or treatment response genes that are differentially expressed due to your treatment, you also have to deal with the background of those genes that, at least in comparing 2 samples, vary quite a bit. Usually to control for that level of background variability, what we're going to need is quite a few biological replicates, and look for those genes that consistently vary with treatment more than they vary between treatments more than they vary within treatments. It's the basic model of statistics, but just looking at this data, we can see that between two different mice, there's quite a bit of background variability in gene expression before we even come to treatment or disease. So it's a noisy system, and you could also say that the variance is over-dispersed. Yeah.
I missed it. How many mice are in?
This is just two mice, one to the other.
One is on the.
Oh, it's X versus Y.
One is the,
Yeah. Yep. Y1 versus Y2. Mouse 1 versus mouse 2.
So how much does it get better usually if it's 5?
A fair bit. If you bin them, yeah, you'll see a reduction in the amount of variability. And that's one way to do it. But if you're going to, well, you would save money on the chips if you mix the 5 mice together for one RNA sample and they treated 5 mice together for the other. But if it's a lot more expensive to grow the mice and extract the RNA, you'll get more value out of running separate chips for each one, because then you more accurately estimate the standard deviation for each gene. Whereas if you only have 2 treatments, then you have no replicates, right? So you don't know how accurately you're measuring each gene in your bin of 5 mice. So there's arguments pro and con, but I like the idea of being able to estimate the variability. Okay, so normalization is a method that can control for many experimental sources of variability, which are systemic, meaning they affect pretty much all the genes on your array together. Not gene-specific. Normalization can't do very much about gene-specific effects. So usually what you end up doing is you end up comparing the same gene from one array to the other, and in that case you're hoping that the gene-specific effects cancel. Say the GC content of the probes will be the same on 2 chips where you're looking at the same gene. You can't control comparing gene A versus gene B. So even though you have a much higher expression level for gene A than B, you fundamentally can't know whether that's a bias based on the detection system. There isn't any way to control for that realistically. From a very simple approach, you could say, okay, we want to make the average brightness of each chip to be the same. This just makes sense, right? I showed you a fuzzy green picture with lots of spots on it, and you could imagine that the strength of the fluorescent reagents, the exact temperature, the flow of electric currents, the focus of the detector could give you a different average brightness, which would tend to make all of the genes more highly expressed on chip B than chip A. So you could divide by the median brightness for each sample. So then you have each sample's relative brightness compared to the average brightness of that chip. That takes care of that. And in fact, it may not be the greatest idea to divide all the samples by the average brightness of the chip, because I showed you that the gene expression values are logarithmically distributed, and very, very high numbers are going to have are going to be affected differently than very, very low numbers. And so there's a method called LOS, which basically breaks the genes into bins along the A-axis, the intensity axis, and it normalizes bins of genes together. Divide this by the average brightness of this bin, this by the average brightness of this bin, this. And you could make the bins sort of arbitrarily small. And this tends to straighten out curvy places on that M versus A plot, which is how you end up getting those nice plots that I showed before. So if there's a hump, say, in the fluorescent emission of your dye at a particular intensity level, a low S normalization will tend to clean that up. Many people have tried to inject standards into the microarray. In fact, Affy has a whole bunch of standards. If we go back to this one here, all of these guys that start with AFFX Those are standards that Affy includes on their chip and in their reagent mix. It does not seem to work very well to use those standards for normalization, although many have tried. Basically, there's no gene which never changes its expression, and no panel of genes which change their expression coherently and consistently across all samples. So if you normalize by one gene or by a panel of genes, you're always going to introduce artifacts and essentially cancel out real gene expression changes that happen to be moving in the same direction as your control genes. Even if the control genes are, in the case of the Affy genes, are external to the system. They're synthetic, or they're genes that come from different species that you've put on the array and then you've put into the hybridization mix. Even still, it doesn't seem to work well. Yeah?
So we are assuming here that multiple experiments have the same amount of the RNA.
Yes, that's a huge assumption. Good point. I was getting to that. We are assuming that there's the same amount of RNA per cell. We can normalize the average brightness of the image, right? If we happen to have loaded different amounts onto the different chips, it'll change the fluorescence systematically. But if instead we have like quiescent cells versus actively metabolizing cells, and then we do that normalization, we cancel the effect of per cell RNA expression going up. We have to assume for all of these statistical methods that the overall amount of RNA per cell is constant, which is, of course, not always true. So that's a weakness of all gene expression-based methods, unless you have some post hoc way of calibrating back on a per cell basis. You have some control that you completely believe in that tells you how many cells were present in each RNA sample. That's a tough one for this system.
But to a certain extent, so for example, in qPCR, you normalize to a gene that doesn't really change expression too much in samples. Can you sort of find that? Like when you have
It's been tried, but when you're looking at global RNA expression as opposed to the expression of just one gene that you're calibrating in a system, pretty much any control introduces some bias. That's just what's been observed.
is that just the nature of the technical variability of microarrays, or
RNA-seq works the same exact way, and the same exact things have been tried and rejected. So yeah, you could do it that way, and there are some experimental systems that rely on that type of calibration, but it doesn't have good empirical validity. And in fact, what you can prove is that when you use that system, certain real experimental differences are canceled out. All right, I'm going to talk a little bit, not hugely, about a nice improved system for generating gene expression, per gene expression values from the Affy cell chips. It's called Robust Multi-Chip Average, RMA. Mostly I'm going over this because I have you do it in the homework, not because I think that you ought to understand this math, which was kind of fluky and developed very specifically for this system. But this PM here is the perfect match value. They're ignoring the mismatch value. And another thing that they do is they do not normalize each chip or each scan one by one, which was the prevailing system before this method was developed. Instead, they take the complete set of chips that you're going to use for your experiment and normalize across all of them simultaneously. So that's why they have this I value and a J value. So that's this same probe across all of the chips. What they assume here is that most genes are not differentially expressed. That is an assumption that's inherent in essentially every normalization method. So if your experiment is so dramatic that you cannot say that most genes, like 75% of genes, are not changing expression between the two treatments, then what these normalization methods will do is they will cancel out real differences. They're assuming that the bulk of genes are not changing. And that's what this median polish part means here. This is like shortcut for a whole bunch of magic math that's not on the slide. So they're basing the normalization on the large group of genes in the middle that have low variance across all samples. And then since some of those will be expressed at low levels, some at medium levels, and some at high levels, yet not changing very much from sample to sample, we can use the aggregate of them to draw a line and then make linear regression on all the other samples to that line. So that's what median polish means. The median expression at different intensity levels for all the genes. If you have lots of genes differentially expressed at one expression level, that's going to screw this thing up. I didn't put the UIs in the AJs. Well, the E is an error value that just represents variance that's uncontrolled, as opposed to variance across treatments and variance across samples, which are these other two. And there's also a background subtraction, which is sort of present in every sample, or present in every probe. So you got to be brighter than some minimum amount in order to make it worthwhile. Yeah.
So the background is per sample?
No, it's actually per probe.
Oh.
Yeah, because there's an average background level in the probes. Or it may be even generalized across a whole bunch of things that are expressing at the same level. But you can't pay a lot of attention to stuff on an array that's basically at the same level as this generalized glow that's going on.
So the background is per chip when you're scanning.
Right, but then you're subtracting it from the value of each individual gene. So it's a background here, but this is generating a number for each gene. So per array, per probe. And then it combines all the probes into one number to give you a per gene expression value. So you're normalizing the probes across all arrays and then combining all the probes in one step. But just in a practical sense, they show you the M versus A plot here before applying this algorithm and after applying this algorithm. And obviously, it cleans things up a lot. It reduces the variability quite a lot. And the most variable stuff is down here at the sort of least reliable end of things, the very, very low values. Whereas in the middle area, probably those things that differ a lot from the median are those that actually show real gene expression differences.
So the x-axis.
Again, it's intensity to the right and fold change to the up and down. So you're seeing things above the red line are fold changed up, and below the red line are fold changed down. And to the right is brighter, and to the left is dimmer. And so you expect to see some big variability in fold change for things that are barely measurable. Sometimes they show a measurement, and sometimes they don't. Or sometimes they show a measurement above background, and sometimes they don't.
Silly question. So because you're splitting you do two chips, one treatment, one control, so the actual probes on those arrays also have a dye.
Also have what?
A dye.
No, the probes are just DNA sequences. The samples are labeled. And you don't just have two. Hopefully, you have several replicates of treatment, several replicates of control. So this is This takes all of those together in one normalization. The nice thing about this system is you could do it with different sets of samples, right? You can mix and match, renormalize a different batch that had, say, a different background because they were collected in a different lab. Doesn't perfectly control for within sample bias, as we've talked about in statistics, but it makes them comparable. And that's the design of the Affy system, is that you don't have to pre-design your experiment. You can take individual chips, put them together into an experiment, and make a comparison this way or some other way. So the first thing you'd like to do after you've got normalized data sets is ask a simple question: are the treatments different? Rather than jump immediately into the differential expression analysis, you'd like to know, do I have a reasonable expectation of finding differentially expressed genes? One of the simplest ways to do that is a clustering of the samples. PCA, MDS, hierarchical clustering. What you would like to see is that similar treatments cluster together, not mixed all together in one batch. And even worse would be some of one treatment in one batch, some of another treatment mixed in with others. In particular, what you don't want to see is a huge batch effect, where all the treatments that were done on day A are here and all the treatments that are done on day B are there. And it's going to be very hard to disentangle your sample, your treatment variance from the batch effect of whatever happened in the lab. If the treatment is responsible for differences that are visible at the gross level of clustering of all the expression, then you have good confidence that when you go in with statistical methods that test each gene for differential expression, the ones that you find with high differences are responsible for the gross effect of the genes of the treatments clustering separately. So these are the genes that are primarily driving the difference in expression patterns between treatment and control. If there are not significant overall differences, then you have to worry that the list of genes with larger fold changes may only reflect random variability and may not be reproducible if you did the experiment again. You would find a different list of genes. And if you did careful false discovery control, you might lose all of those differential expressed genes. They may not exceed the multiple testing threshold. OK, so you need replicates in order to use statistical methods. It's not just the genes with twofold increases, which is how people were publishing microarray experiments in around 1998. It's like, OK, we saw these 17 genes with twofold change in expression from treatment to control. And we did, one sample of each. And eventually, in a lot of fields of science, it becomes more rigorous. And people require more evidence and statistical validation for those conclusions. You have to do your experiment on multiple treatments and show an appropriate statistical test that proves that the changes are real and not just an artifact of noise in your data. For this type of data, for a number of reasons, but most strongly because of small sample size, non-parametric methods may be better than parametric methods. So a parametric method would be a t-test. But if you have only 3 or 5 or 2 replicates of treated and control, then you can't assume a normal distribution of measurements. And you can't accurately calculate the standard deviation of those measurements. You also can't accurately assume that the standard deviation of your treatment samples is the same as the standard deviation among your control samples for this gene, for each gene. And those are all assumptions that are more or less required in order to use a t-test. So there's a variety of non-parametric methods that have been applied to gene expression data. Another thing about gene expression data that we know is that it is over-dispersed, meaning there's more variability in these measurements than you would see in a typical normal distribution. They're spread more widely around their mean. Meaning if you sample a few times, your chance of getting widely dispersed values or values all to the left of the mean or all to the right of the mean, are pretty high. Or the mean of your few samples may be far from the true mean of the population if you had sampled infinitely deeply. You get very highly variable data. Okay, also in a microarray experiment, each gene or probe set is effectively a separate experiment. And you're going to do a separate statistical test on each gene. The problem is if you just had random data and you did 20,000 statistical tests with a p-value of 0.05, you will definitely find some genes that have differences greater than that threshold, just by chance in a randomly distributed data set. Those would be what we would call false positives. You don't want that, so we have to have some control for false discovery, as we discussed in the past couple of lectures. Another problem, which is an assumption that we would make in this kind of approach, is that the different genes are not truly independent, right? We're measuring them and testing their expression separately, but in fact we know that there's a lot of coordinated expression, that the genes are moving up and down together, or in some cases opposite each other. When this one goes up, this one goes down because it's a negative regulator of that and a positive regulator of itself. So some of the assumptions for multiple testing control methods are also not valid on this data. In other words, if you use the Bonferroni, which assumes that every test is truly independent, you're going to over-control and wipe out a lot of true gene differential expression. So you're going to increase your false negatives way more than you need to, and in fact destroy the power of your test. So statisticians call false positives a type 1 error or a false discovery. Obviously, the false discovery rate should be smaller than the number of real differences that you find. Typically, biologists tend to be comfortable with false discovery rates in the area of 5 to 10%. Sometimes you could push it as high as 20%, so that you got 100 genes and 20 of them are probably false, but the other 80 are probably good, statistically speaking. So that's sort of the comfort zone where we operate. It's not really hard and fast proof, but it's a reasonable way of dealing with data where the variability is not well understood. You also, when you do a false discovery rate, that's an estimate, right? You don't know for a fact that you have this many false genes. It's just based on the variability of the data and the power of your test, this is the number of false discoveries that we expect to see at this significance level. So that brings us to SAM, significance analysis of microarrays. Again, a method that was specifically developed to deal with this exact data type and the kinds of data sets that biologists were generating. It's available as an R package, which is how we're going to use it. And it's also an Excel plugin. It's very widely used and highly cited. It uses a permutation-based approach. And I'm going to attempt to explain this, although I strongly recommend you follow up and read some of their papers, their website, etc., if you want more detailed explanation. So just a general overview, right? You have gene expressions. By scale, they mean normalize. You're going to calculate a statistic, which they call D, which is very similar to a t-statistic. It measures differential between treatment A and treatment B.
Is that the same as Cohen's d?
Their own D. They made it up. Their own D. You're going to estimate the difference between treated and control. I said that. Now you're going to permute the sample labels. So you have 6 replicates, 3 treated and 3 controls. You compare the treated versus the control, and you calculate a D. Now you mix them up so that you've got some treated and some control in the 2 bins. And again, you calculate the D. You do that again and again. There's actually only a finite number of mixes that you can make before you repeat yourself. If you have a few more samples, like 5, you can have a lot of good permutations. If you have only 2 samples of each, there's very few permutations that you can do. You're going to calculate the D for all the permutations and calculate the average D. That's the expected difference. And then you're going to see, does your actual difference exceed the expected difference of the permuted samples? That's what it's all about. There's a delta. Oops, there we go. This is what I just said. Oh, you're going to plot the observed differences versus the expected differences for every gene. Again, the assumption is that most genes are not differentially expressed. So, oops. So the bulk of the plot should be fairly close to the diagonal. In other words, the observed difference should be pretty close to the average of the permuted differences. Or the observed D statistic should be pretty close to the average of the permuted D statistics. Up here at the high end, you're going to see some genes that have higher differential expression between the 2 groups than you would expect. And then there's this particular statistic is symmetrical, or at least it measures downwardly regulated genes as well, where the differentials between B is greater than A. So A is greater than B is up here, upregulated genes. B is greater than A down here, downregulated genes. For most genes, the difference is fairly close. OK. So this is the D statistic. It's very similar to this T statistic, except they add this little, what they call a fudge factor. I mean, they actually say that in their paper. What it does is it increases the measure of variability for very low expressed genes and pushes down the D statistic so that you're less likely to find differential expression in genes that are expressed at a very low level. Well, this is a number that it's a constant. It's in their formula, so yeah. And this thing was built for Affy cell files, so I think that number may have something to do with the range of values that you typically see. I showed you from 0 to 40,000. So, stuff that's less than 10 is low on that scale. Probably things that are less than 100 are low on that scale. I used to delete all the gene expressions lower than 100. OK, I said that. This is how we do the permutations. I said all of this. Oh, the FDR. You can also calculate a false discovery rate from this same graph simply as the number of genes that exceed a given cutoff, meaning a difference between observed and expected, in a permuted sample, or an average of permuted samples. So when you permute and you compare the D statistic on this one permutation versus the mean of all permutations, it shouldn't be significant, right? But occasionally some of them are. You can rank all of those differences on the same scale and say, OK, we're going to cut at the 90th percentile. We're only going to pay attention to differences that are greater than 90% of the differences we observe with the permuted samples. So, or 95%, or whatever cutoff you want. And in the software, especially the Excel version, they have this little slider. And what you're sliding is the D, the difference, which is an arbitrary thing. I mean, it's a 2 or a 3 or a 5. But when you slide it a given amount, it tells you what the FDR would be. It tells you the number of significant genes at that level and how many false discoveries there are. In a good data set, you can slide it to the point where you have, only 5% or 1% or less false discovery and X number of differentially regulated genes up and down. In a bad data set, you get once you slid the delta up to the point where less than, say, 10% of your discovered genes are false, you may have 0 significant genes. So that would be a bad data set where the effect of the treatment is less than the background variability among samples within a treatment. So this is a popular method. It's implemented in actually 2 different R packages. We're going to use the simpler one called SIGGenes. OK. Oh, I'm not keeping track of time. Clustering and pattern detection, data mining, statistical validation, meta studies. All right, I'm going to try and get through this somewhat quickly because it's going to be covered a lot of other times in other areas. We can do hierarchical clustering, self-organized maps, and principal components. A hierarchical clustering like this clusters your samples on one axis and your genes on the other. This was invented by Mike Eisen in a pretty popular piece of software that he just called Cluster. And it's interesting. I mean, certainly the clustering of samples is important for what I said before about when you look at all the genes, do the samples cluster in a sensible way, treatments and controls separately. In that case, if they do cluster separately, those genes which are up, the green ones here, may represent some sort of biologically coherent group of genes. Those genes that are, differentially expressed upward by your treatment, and those genes that are differentially expressed downward by your treatment. That's what I'm going to say about this for now. In a more interesting experimental design, like say a time course, you might be able to track a block of genes that move consistently together across the time course. And so they're green here and red here in different sample groups. So this could become very interesting data exploration tool. Well, this is a piece of software that does a lot of this stuff with a push-button interface. That's all I'm going to say about it. I think the school still has a license for it, but you can do it in Bioconductor. You can do it. I mean, it really is just a push-button interface to the pretty much same tools that we have in Bioconductor. And it's kind of expensive, but I think you can get it for free. This view I particularly like. And in Bioconductor, it takes quite a few lines of code to generate this, whereas in GeneSpring, it's a button push. So you see it much more frequently in people who use this. So this is showing individual genes that change over a bunch of different treatments in some sort of consistent fashion. And you can preload different sets of genes and see how they behave. So it's a nice data exploration tool. Self-organizing map clusters group the genes, they group the samples in specific ways. And I actually don't have the time to talk about the algorithm, so I'm going to skip over it for another day. Classification I'm going to do very, very quickly, but I am going to put in a plug for this tool which automates the process. Again, it's taking all of your samples, applying a particular, it's supervised classification. So you're saying, I want to find genes that separate my treatment from my control. And I want that to be significant, not just a random set of genes that separate treatment from control. So it's class prediction. All right, I'm not gonna be able to go into this because I want to show you some of the other parts of the microarray stuff. This is what I want to focus on, is that there's this tool package in R called Bioconductor, which has received a lot of software development over the past decade or so. Pretty much all biostatisticians who work in this area develop their software for Bioconductor. There's a lot of tools that have very sophisticated methods and fairly simple set of commands to get at them. Unlike Python, where it's much more build your own tools using sort of more fundamental statistical processes. In some cases, Bioconductor has very complex data objects which have methods built into them. So you can get, say, an image plot from a cell file with one command, or pull out the perfect match probes and graph them with one command. So it's worth learning, and also, as you saw from the biostatistics lectures, it's very widely used among biostatisticians, R. And then Bioconductor is a set of pre-built modules in the same way that, say, Pandas or BioPython is for Python. So we're going to mess around with it today. One last thing I want to talk about is the functional interpretation, which is sort of at the bottom of my workflow. Take a list of interesting genes, say differentially expressed, and find their biological relationships. This obviously requires a reference data set of biological knowledge. And traditionally, biology is a messy place where people working in different systems, say fruit flies versus bacteria versus mice versus plants, tend to name genes and functions and even cellular components using different sets of terminology. And in order to do a consistent gene expression knowledge, consistent gene functional knowledge data set, we need one ontology, one set of biological meanings that can apply to all organisms equally. And strangely enough, the biologists actually got together, created a working group, and came up with a consistent set of functional annotations for genes, pathways, and functions. And it's called the genome ontology. The genome ontology labels gene function, and it's at the gene level, although it's probably helpful to think about it at the protein level more than the gene level, because proteins have biological locations. You know, it could be a membrane protein or a cytosolic protein or a nuclear protein. And it's less helpful to think about the gene having its function in the membrane. It's much more helpful to say the protein is actually present in the membrane. So there are biological processes, things like development, cell division, gene regulation, a pore that regulates the movement of some molecule in and out of the cell. Cellular component, which generally refers to a geographical location in the cell, nucleus, cytoplasm, mitochondria, etc. And then there's molecular function, which is at a very micro level. It's a phosphorylase, it's a kinase, it's a structural protein in the actin filament. It's whatever it is at the molecular level, very small scale. And they have about 1,000 or so general categories. But an interesting thing about the structure is it's called an acyclic graph. It's hierarchical, but the same protein can be present in several different hierarchies, because that's the way biology works, right? Sometimes proteins are involved both in cell division and in the regulation of metabolism. They could have multiple targets, et cetera, et cetera. So it's possible to annotate a given protein in many different functional hierarchies. There's a really nice public website that is very, very widely used for functional annotation called David. I actually forgot what David stands for. It has a few basic features. One of them is it has a complete list of gene identifiers from several different systems. It has GenBank, it has EMBL, it has SwissProt. It also has Affymetrix gene IDs, and it has Illumina gene IDs, and a few other things as well. So given an experimental system where you've done some statistics, produced a list of interesting genes, you can paste that list right into David. David will internally translate it to a consistent set of identifiers, and it will tell you if any of the identifiers in your list for some reason are not mapped internally to its own gene ID list. And then you might say, why is that? Maybe I incorrectly identified these genes, and I'll try a different ontology for my list of gene names. Or I'll match them to names X somewhere else and then come back to David with new names that it can interpret. Every gene ID is matched to a function in the ontology list. That's a fairly simple one-to-one matching and takes very little compute power. And then, given your list and the matching categories that it pulls up, it tells you whether that set of categories is enriched at any level compared to a random set of categories. It groups the significant categories into functional clusters as well. I'm not going to dwell on this, but it basically uses a variant of the Fisher's exact test, although functionally it ends up being a lot like a hypergeometric test. It just says, all right, you gave us a list of 40 genes. Oh, you gave us a list of 300 genes. 3 of them are in the p53 signaling pathway. We know that the human genome has 20,000 genes, and 40 of them are in the p53 signaling pathway. Is 3 out of 300 more enriched than 40 out of 20,000? And is it statistically more enriched according to the Fisher's exact test? They're actually more stringent. They subtract a degree of freedom, essentially. They do a -1 from your gene list to make it more stringent because they find too many false discovery otherwise. So they call their method ease. And that's all it is it's 3 minus 1 divided by 300 compared to 40 divided by 20,000. All right, last thing is public microarray databases. If you want to publish your gene expression experiment, the journals have agreed that the data must be made public. There are a number of gene expression databases, but the most widely used one here in the US is run by NCBI, and it is called GEO, Gene Expression Omnibus. And it will take the cell files, which is the gene intensity, perfect mass, mismatch data files that are generated by Affy. It'll take gene expression files generated by a lot of other manufacturers. It'll also take RNA-seq data in FASTQ form. It will also take your analyzed data, the spreadsheet that you've made of normalized gene expression values and the list of fold changes and p-values that you've calculated. And it will allow you to annotate according the methods that you use to generate that analyzed data. So raw data, spreadsheets, some text. Metadata is required, obviously. What the samples are, how they were treated, who you are, how the scans were made. If you're uploading data files, you obviously have to say what's the platform, meaning what was the chip that you used. And that should point to some sort of set of gene definitions. Usually, if you're using an Affy chip, you can just point right back to the manufacturer. Or actually, in the case of Affy chips, Geo already has definitions for all those chips, and you can just specify that chip from kind of a pull-down list. It has a very simple data structure: submitter, platform, series, and sample. Sample obviously is an individual cell file. A series is a bunch of data samples that are linked together in an experiment. So a series is an experiment. This is a nice, simple data structure. And it also has a decent, although not horribly accurate browser. So you can go in and look for experiments on gene expression in pancreatic cancer or hypoxia treatment of yeast or whatever your particular interest might be. Unfortunately, the browser is relying on the description that the investigators have loaded, which, as we know, scientists are not horribly consistent in their use of English text. So it's only as good as it is. You might go reverse, browse PubMed, find experiments that have been published, then use the Geo ID that they've included in their publication to come back and exactly find the data set that they used. Now that we require Geo for publication, you've got that two-directional link from the data set to the paper and from the paper back to the data set. Yep.
I would like to add that after spending a lot of time trying to submit these databases, they don't make it horribly straightforward.
It's,
So it is possible for people to mess up
and have
input mistakes and
Oh, mistakes for sure, and also lack the data that's supposed to be there. I mean, you can go to a published data set, go to the Geo project, and not find what you expect because the curation is kind of not very tight. You know, you will have created a project, you will have loaded some of the required fields, and so the project exists, but you fail to correctly load some of the data, and nobody will ever know until years later you try and download the data and it's not functional for some reason. True, but nonetheless, there's a lot of data here, and it does get reused in other experiments fairly often. It's very exponential, the distribution. A few data sets are used a lot. Most data sets are never used again. That's true for scientific publication citation rates also. A few everybody uses, most nobody uses. OK, they also have a full change browser, which is kind of cool. You can type in a gene name, and it'll show you all the projects where that gene is differentially expressed. Now, most of those projects will have 2 replicates, and the differential expression is, completely bogus. But every now and then, you will find something really interesting about your gene in this other experiment where you never thought that gene would be differentially expressed. So here's a classic example where you've got 6 replicates and some genes showing nice differential expression. And this is all done on the Geo website. You don't actually have to download all the data, renormalize, and run your own analysis. So it makes it fairly quick to go through some data.
Now, when you type in your gene name, you have to type in, because some data sets, the only genes that they publish like a gene ID or like an ensemble gene ID or something like that.
Well, this is a GenBank tool, so GenBank IDs are preferred. Universal gene IDs from Hugo will work for humans and mice. Other species, who knows?
So they take that data, or so they curate the data in a way that, even if the data set is uploaded with a different set of gene IDs.
Well, a lot of them are uploaded as cell files which have Affy gene IDs, and they have an internal mapping of Affy gene ID to GenBank ID. It's not perfect because the Affy gene IDs are highly redundant, and some of them point to non-existent genes. So I can't say it works perfectly, but that's the idea. And I have seen some interesting data discovered this way. So like for well-known genes, RAS or whatever, is probably going to work pretty well. Oh, that's what I'm showing here, geo profiles. So this is typing in a GenBank ID. You see up here at the top, NM-whatever. It's 06953. And then this is the list, 434 experiments that have differential expression observed for this gene ID. And then you got to sift through them one by one. There's not like a tremendous level of good filtering to say, minimum of 6 replicates, minimum of this fold change. That's not really available. So, this set looks good, but this set maybe not, because there's 1 sample with high expression and 2 with low, so that's kind of lame. Okay. Right. Microarray experiment, data formats, QC, normalization, differential expression, functional enrichment, and public databases. Right. Next week, we're going to start talking about next-gen sequencing. I used up my time for the R introduction. It's already way late. It's on the website tomorrow when David gets back. Sorry. I really didn't. Well, you know why? Because you guys asked questions. That's why. I've gone through this whole lecture in 70 minutes. No, the questions are good. That means that you